Alright, so here we have some terms that we need to make sure that we know for looking at this unit. So DNA, chromosomes, genes, alleles. Before I look at the definitions, let's just look at how the DNA is composed. That will help us to talk about the different definitions. So the nucleus, as we remember, it is where the genetic information of this cell is housed, and inside of there you have these chromosomes. Now, as you can see, the chromosomes are actually very tightly wounded DNA or DNA molecules. So when we look at this DNA structure here, it's a double helix shape, and that wounds itself around these proteins, and then those proteins wound themselves around each other, and eventually you get this very condensed chromosome shape. Now, normally when you see the chromosome in the cell at the beginning, it's just going to have one side, and only when it's starting a new sort of division of the cell would it replicate and form that other pair. And that is known together, those are known together as sister chromatids. So if you were going through and you saw sister chromatids, that's what those are. But in the beginning, there would only be one single one inside of the cell if it's just in its regular everyday state without trying to undergo mitosis or meiosis. So the definitions, let's look at those. So DNA stands for deoxyribonucleic acid. It's a chemical which holds our genetic code or instructions, whereas chromosome is a thread-like structure made of DNA wrapped around these histones, which are the proteins, so that the DNA is packed closer together. And then gene, it's a segment of the chromosome which gives instructions for a particular characteristic. So you can have a gene for hair color, a gene for hair texture, a gene for eye color, etc. And then allele are all the alternative forms that you can have of the gene. So for example, you could have the gene for eye color being brown, green, etc. So as I was saying earlier, this is how your chromosomes would look in their everyday state. So as we know, we have 23 pairs of chromosomes. The third pair, sorry, not third, the 23rd pair is the sex chromosomes. And as you can see, the X is much larger than the Y, and all of the other chromosomes, as you also can see, they tend to all be similar shapes, right? When you see those similar shapes and sizes of chromosomes, it means that they're carrying the same types of genes. So for example, this red section here may carry the gene for hair color on both chromosomes, but on one hair color it might be brown, but on the other one it might be green or black or some other hair color. So these are known as homologous pairs. Whenever you see homologous, it means same. It's a pair of chromosomes of the same type and shape. This is just another way of looking at that same thing. So let's look at mitosis. So this type of division is very important in growth of your cells and in growth of your tissues. And the reason why it's so important for this growth, not only growth, but also repairing the cells if there's any damage, or if there's any damage of your tissues, is that it produces identical daughter cells. So when the cell divides, it makes an identical copy of itself. So as you can imagine, therefore, it's also very useful in asexual reproduction, where you have to make the same types of organisms or the same types of tissues For example, this plant here is something you might see, bryophyllum, and it produces these little identical leaves on top of the margin of the leaf, and each of those plants is going to end up being an exact clone or replica of the parent. Starfish also do similar thing, where they produce identical copies once species break off. Now, something else to keep in mind is that when the daughter cells are made, the chromosome number is maintained. And what that means is that if you had 46 chromosomes, such as us in our cells, 46 chromosomes would be in the daughter cells as well. And this is known as the 2n number, or diploid. If you think about di for 2, diploid number. This is going to be important when we come to meiosis, which actually has half the number. So, due to the daughter cells being clones of the parent cell, this causes the tissues resulting from the cell division to be identical to the parent. Now, here are some representations of that. Let me see which one I think will be easier to understand. Let's look at this one. So, remember I said in the beginning, your chromosomes are going to be these single strands. You see them there. Then, once they start to prepare for cell division, they're going to replicate, and we'll see why this is important. You should not be asked about the stages, but just in case, this one, this stage here is known as prophase. This is before any sort of cell division happens, and each chromosome is just creating an identical copy of itself. And as we said, those are called sister chromatids, and they're attached at something called the centromere. Then, what you see happening here is that they're lining up along the center or the equator of the cell. You can see too that the nuclear envelope fully disappeared and allow them to line up straight down the cell. This is known as metaphase, or they're meeting at the center of the cell. You also would see that there are some fibers attaching to them at the center here, and what this will help to do is it will attach to them and be able to pull them apart from each other in the next phase, which is anaphase, which means pull apart. A for apart, M for meeting in the center. Now, what you're going to observe here is why the replication was important. Once these daughter cells are made and you count how many chromosomes were in each daughter cell, 1, 2, 3, 4. 1, 2, 3, 4. You would realize that you end back up with the same number that you had in the beginning. If you did not replicate those chromosomes at the start, you realize when they lined up, you might just get 2 in each, which is not what we want. Because we want, at the end of mitosis, to have the exact same number. It's supposed to be a clone of the parent. Okay? Now, how does this differ from meiosis? So as you can imagine, meiosis, you're going to have unidentical daughter cells, and it's used in sexual reproduction, and you produce gametes. Now, as I was saying earlier, because you have half of the chromosomes, remember when we spoke about gametes always having half, so our egg and sperm cells each have 23 chromosomes, so that when they come together, you get the full set of chromosomes that is needed for a human being. So the diploid number of chromosomes is halved, and this is called haploid, or N. So it's no longer 2N, but just N. So halving of the chromosome number for gametes allows the diploid number to be restored during fertilization. And fertilization allows half of the genetic information to come from the mother and half from the father. This word here, variation. how are we going to get differences being introduced into these daughter cells? There's something interesting called crossing over and independent assortment. The one I want you to focus more on is for tomorrow is crossing over, and I'm going to show you what that is. So let's look at this same one here. So again, in the beginning, we have the chromosomes as they are, not doing anything. Once the cell signals that it wants to replicate or it wants to divide for meiosis, the chromosomes are going to duplicate themselves again and attach to each other. Their sister chromatids are going to attach. And then something interesting happens here. Instead of lining up directly down the center, just one after the other, you realize that they're pairing up. So they're pairing up, their homologous pairs are pairing up. Remember what I said homologous pairs meant? It's when you have the same type and shape chromosomes coming together. So one was from father, one was from mother, carrying the same types of genes. And again, they're meeting at the center, so it's actually also still called metaphase. It's called metaphase 1. Then, in anaphase 1, you have them again pulling apart. What goes over here? 2 chromosomes. Over here, 2 chromosomes. How many chromosomes did we have at the start? 1, 2, 3, 4 chromosomes. So we already see that half the number of chromosomes has resulted at the end of the first division of this cell. Simply because they paired first and they didn't line up down the center and allow for the chromatids to separate. Instead, the pairs fully separated from each other, only resulted in 2 in each cell. Then, what we have coming up again is another set of metaphase where they meet again at the center. It's metaphase 2. And then as you can see here, now the sister chromatids are the ones that are pulling apart from each other. So this is phase 2. And at the end of it all, you see that we have our individual chromosomes, just like in the beginning. But again, how many? Half and half. So we only have 2 here, 2 here, 2 here, 2 here. In the beginning we had 4. Now we're crossing over. Where is this happening? At this stage here, when they start to pair up, the homologous pairs, they actually would also swap DNA with each other and start mixing mother and father's genetic code, so that you would have pieces of DNA coming off from either one and going into the different cells and creating a lot of different types and variations of the sperm or egg that was formed. And in plants, this meiosis would also occur, but it would take place for the pollen, which is the male part, or the ovule. So in the next video, I'll just go over some past paper questions related to these particular parts of the topic.